All right, so last talk for today, since we have a short day, it's my pleasure to introduce Thomas Stiehl, who is not only a mathematical biologist, but also has an MD degree. So it's a rare breed of mathematical biologists. And he works at Aachen University and also Roskilde, and he's going to talk to us about evolution in the hematopoietic system, right?
Thank you, Dominic, for the kind introduction. Thank you so much for giving me the opportunity to talk here. So I am talking about joint works with different people. So still Anna Marciniak from Heidelberg, Christoph Lutz and Anthony Ho from Heidelberg. Then Wen Wang, who actually is now at Fudan University, and then together with Aachen people. So Rebecca Schneider, Wolfgang Wagner, Shenju Chu, Shoujie Wang. So the latter two are my PhD students. And of course, I also have lots of common work, as Morten already mentioned, with the people from Roskilde. But here I try to focus a bit more on the work I'm doing in Aachen, not to have too much overlap. So why am I considering the hematopoietic system? So I think we all agree that the hematopoietic system is kind of paradigmatic example for stem cell processes during regeneration, homeostasis, and also diseases. And actually, the first part of my talk is about acute leukemia, so mostly acute myeloid leukemia, which is still a very severe disease with a highly toxic treatment and the majority of patient relapses. So if you compare the progresses which have been made in pediatric leukemias, they are much stronger than the progresses in the adult myeloid leukemia. So there is still lots of room for improvement from the clinical side. So I think Morten has already nicely introduced the blood forming system. We all know that blood formation takes place in the bone marrow and is hierarchically organized with hematopoietic stem cells at the top, giving rise to progenitors and precursors. And in the end, there are functional mature cells. Most of them do not divide anymore, except some subsets of white blood cells. And these functional cells have specific jobs they perform in your body. So I now consider a very simple way to model it. So I mainly focus on the production of granulocytes. So I just cut out one part of this tree and describe hematopoiesis as a finite sequence of discrete maturation steps where all the cell types are characterized by a set of properties. So for simplicity, I neglect death of the immature cells, and in healthy homeostasis, I assume the death of the white blood cells, so more specifically the neutrophil granulocytes, so the largest subset of white blood cells in the adult human. I assume their clearance rate to be constant. Then each cell type in this hierarchy is characterized by two parameters, namely the proliferation rate, just quantifying how often on average a cell divides per unit of time. Then for the mature cells, the death rate, and then the so-called fraction of self-renewal. So usually in this process, cells have to take decisions whether they differentiate or whether they self-renew. And if you imagine you have stem cells dividing, then a certain percentage of their offspring might be against stem cells, and the complementary fraction might be more mature, so namely early progenitors. And to quantify this, I use the fraction of self-renewal, which is basically, if you imagine you have 3 stem cells giving rise to 6 offspring, and 2 of them would be against stem cells, then the fraction of self-renewal would be 2/6. So this is what we use to describe the differentiation process. And this model can basically be represented as a system of ordinary differential equations, very easy, which takes care of all these fluxes I have just introduced. And as we all know, the hematopoietic system is tightly regulated. For example, in response to injury or blood cell loss, but at the same time also in response to infection, hypoxia, and other stimuli. So we have to bring in this model feedback mechanisms. There are many different ways of doing it. We have studied plenty of feedback mechanisms. So I'm now showing you a very simple one. For the feedback mechanisms we use a quasi-steady state approximation. So if you think about stem cells dividing maybe once to twice per year, and then you look at the change of cytokines, so the feedback signals, they are very fast. This is, for example, known after blood cell donation, after blood platelet donation, in response to trauma or injury, that the concentrations of the feedback signals reach new values within minutes to hours. And in contrast to these relatively slowly dividing stem cells and the long time scale of disease evolution, the processes in the signals are relatively fast. So what we usually do is we describe signals by an additional equation, which is, for example, a constant production. So you all know EPO, which is the main signal of red blood cells. But what we look here is GCSF, for example, so the main signal for the granulocytes, which under some additional assumptions is more or less produced at a constant rate and then eliminated by receptors on the surfaces of the cells. So this is the idea. And if you assume that this process is fast, we obtain a first order Hill function, which we then use to describe the feedback signal. Then the question is, what could be the actions of the feedback signal? So there are different versions which we tested. It is relatively well known that the feedback signal increases the proliferation rate. So a factor of 4 or 5 in the more immature compartments, for example. Then another important factor which we saw when we modeled bone marrow transplantation is that the feedback signal increases self-renewal. And then basically we put this in the model equations, just we replace the respective parameters which become time dependent by these Hill functions. So this is the main idea. Yes?
So the feedback signal, the natural cells fit all the previous compartments, not like next compartment to the previous.
Yeah. So I mean, this is at least
simplification, or is like what
there are several simplifications. So what you can do is you can look at cytokine receptors on the cells. And what is known is basically that receptors for GCSF are more or less on most of the cells, but the higher the degree of maturity, the more receptors they have. So this is basically the reason why the signal here. So this is basically the reason why the signal here depends just on CN. And we have other versions of the models where you can have, as you said, feedback to the directly previous compartments. So for many of the models, it does not make such a big difference. We also have versions where we have a local feedback on the stem cell and then the feedback from the periphery to the progenitors. So there are slight differences, but all in all, the output is qualitatively comparable.
And there's work like Natasha Roberts, right?
Yeah.
Looking at different architecture and
exactly.
Colon cancer,
exactly.
Compatible with the data, right? And she
exactly. Exactly. So what the criteria we usually use to choose the feedback is, first, we want the healthy state to be stable. And second, we want to have realistic repopulation dynamics after large perturbations.
So these feedbacks, are these positive feedbacks? You mentioned that they increase the proliferation and increase the self-renewal.
So exactly. So if there is a lack of cells, then proliferation increases. So in this sense, it's negative. So the more cells you have, the less the cells, the immature ones, do. And then the question is, as I already mentioned, how can we study such kind of feedback mechanisms and what might be scenarios to see if our model is reasonable? So one such scenario is bone marrow transplantation, because of course what you want to see is a scenario where the system is far away from the steady state, because there the feedback mechanisms take action. And bone marrow transplantation is such a scenario. So bone marrow transplantation is used as a curative option for many hematologic diseases. And the idea is you eradicate a host's marrow using chemotherapy, sometimes supplemented by radiotherapy, and then you infuse stem and progenitor cells to the bloodstream, they home to the bone marrow, where they repopulate the system. What we are looking at mostly is the so-called autologous bone marrow transplantation, where the cells which are transplanted have been harvested from the same person. So we don't have autoimmunity issues or graft versus host issues due to different immune phenotypes. And this is basically what we use to test if our feedback model is realistic. And what we also use to calibrate the model are basically equilibrium cell counts of the different stages in bone marrow, which are more or less known. For some cell types, there exists estimates on proliferation rates. So this gives us constraints on the parameters. And then we use the bone marrow transplantation scenario basically to calibrate the time-dependent part from the signal. And then we end up with a model which can recapitulate different dynamics of white blood cell recovery after bone marrow transplantation. So what you see is if you eradicate the host's marrow, the cell drop, and then after the transplantation, they recover. This is what we observe. And the model is basically able to recapitulate this and also to recapitulate the dose-response relationship of transplanted cells and time to recovery. And this is basically the model which we use as a point of departure to describe different diseases. So what we see from this model is that the regulation of self-renewal is very important to have quick recovery after bone marrow transplantation. And what we also see is that this relatively simplified feedback, only depending on the mature cells, is at least sufficient to capture what we observe in routine clinics.
Sorry, could you say how many steps there were?
That's an excellent question. So we have different versions. The simplest is 2, and the most complicated is 8, where we really go into all these different steps of white blood cell formation. This is for the 8-compartment model. Okay, then the question is, how can we extend such a model to account for leukemia? So leukemias are diseases mostly of the white blood cells. And what we observe in leukemic patients is that healthy blood cells drop. So this means there has to be some kind of interaction between the cancerous cells and the healthy cells. And we do this basically by adding a cell lineage. So we have then the healthy cells in the model and at the same time the leukemic cells, and we let them interact. So there are different ways how they can interact. We tried multiple possibilities. One possibility is that there is a kind of competition for the feedback signal, which is justified by the finding that in many patients, leukemic cells express receptors for the signaling factors, and cells and graft. So if you transplant the leukemic cells to mice, they grow better if the mouse produces the human signaling factors. So this means that at least in a subset of patients, the leukemic cells also depend on the feedback signals. And this is what I will assume for this talk, but it's not the only way to model it. And then what we basically have is we have systems of differential equations, one subsystem for the healthy cells, one subsystem for the leukemic cells, identical to the one I have presented before. And the coupling is basically through the feedback signal, which depends on the healthy and leukemic cells in the last compartment. So this is the idea. And then one question we asked is we now go back to keep the things simple to a 3-compartmental model. So 3 compartments for healthy cells, stem, progenitor, mature, and 3 compartments for the leukemic cells. So stem, progenitor, and post-mitotic. And then one obvious question to ask would be, what is the impact of stem cell parameters and progenitor parameters on the pace of disease progression. So what we then did is we calibrated the healthy part of the model, and then we systematically, as kind of sensitivity analysis, varied the parameters of the healthy cells. So here you see an arbitrary time scale. And then what we basically see is the disease progression is fastest if stem cells proliferate quickly and have a high self-renewal. If we then do the same procedure for the progenitors, we see that the sensitivity of the time to disease outbreak or relapse with respect to the progenitor parameters is much smaller. So this is the same time scale compared to the parameters of the stem cells. And this basically means the bottleneck of the system is the number of cells which leave the stem cell compartment. Because then in the progenitor compartment, you have relatively quick division and more or less within certain constraints, a fixed amplification. So this was what matters is basically what do the stem cells do and what is the influx into these amplification stages. And based on this, what we tried then, of course, what now would be an obvious question, can we fit such parameters based on clinical data? So what we used is data at the diagnosis of AML, where we basically knew how many cancer cells are in the bone marrow, and then the patients were treated. We only took those where the treatment was successful, which means where the number of cancer cells is below the detection limit. And what we then did is we observed them. So this is retrospective data from clinical routine and waited until they relapsed. And then at the time of relapse, we basically knew how many cancer cells are in the bone marrow and what is the time interval between end of therapy and relapse. And then the question is, can we fit leukemic stem cell parameters in the model based on such data? And what we see is, of course, the system is not identifiable. So we see that for every patient, we get a one-dimensional line in the two-dimensional parameter space of combinations of stem cell self-renewal and proliferation, which could explain the data. So this is not so unexpected. But if we now repeat this for multiple patients, we see that basically the potential parameter combinations are spread all over the parameter space. And then what one would expect is that the location of the parameters in the parameter space might provide us some ideas how quickly it would take to a second relapse and ultimately to the death of the patient. So we tried this using, in the end, some cross-validation procedures to subdivide this parameter space and then check how the patients survive. And what indeed we see is that patients where the model predicts fast proliferation, high self-renewal, they die quicker in the end than patients where either proliferation or self-renewal is slow. So this is the proof of the principle that the parameters which we estimate somehow correlate with the clinical prognosis.
So those boundaries are parallel basically to this.
Yes.
Time. So there's something like the product of two, which is the
invariant. Exactly.
So you could reparameterize, right, by using some invariants.
Yeah, in principle, yes. So what is our conjecture is basically what matters is somehow the product between proliferation and self-renewal.
But that's what I'm saying. It's
Yeah, it's the
hyperbolas. So.
Exactly. It's more or less hyperbolas, but it's difficult to really get it reparameterized exactly because we have the feedback signals and we have a minus in between. So this is what makes it formally a bit.
But you could write down.
Yeah, exactly. Exactly.
There was a question in the chat, by the way.
Yes. Okay.
How do we identify non-identified? The chat.
Yeah. So what we in principle did is We tried the standard parameter estimation techniques. And then in the end, we saw correlations between parameters. And what we then basically did is we swept all parameters on a fine grid within the physiological plausible ranges, and then checked how far are we away from the data and how good we can reproduce the data. Because I mean, you know more or less a cell can divide not more often than twice per day or one per day, and stem cells maybe divide with 1 to 2 times per year. And this is basically the range we checked. Then the self-renewal is a probability, so it has to be between 0 and 1. And then we basically just see that it is not unique. Okay, so based on this, I mean, this is all on retrospective data, so far not validated prospectively, which is, of course, a drawback. But I mean, a prospective validation is a bit challenging, so this has not been done yet. So, of course, you have to take all what I show you with a grain of salt, because you never know what happens if you do it really in cohorts from different centers and prospectively. So, but what this model suggests is that the leukemic stem cell properties seem to have impact on the disease prognosis. I mean, that's a simplified model, or let's say at least the relatively unmature cells. With this 3-compartment model, of course, you cannot really say, are these the stem cells or the very early progenitors? And we see that basically high self-renewal and high proliferation lead to a poor prognosis. So this is nice. Of course, the problem is it is clinically not useful, because what we need to parameterize this model is data from diagnosis and data from first relapse. And usually, if you want to have a prognostic stratification, it should be available at the time of diagnosis and not at the time of the first relapse. So then the question is, might there be a different way? So what we have done so far is we took data mostly of the cancer cell burden at 2 time points. So a different approach would be to measure multiple cell populations simultaneously at 1 time point. And this is what we tried. And to get our hands on this, we refined the model and we accounted for the stem cell niche. So there are, in fact, multiple stem cell niches. We have a very simple setup with only one stem cell niche, which is shared between healthy and leukemic cells. And the assumption is the cells need contact to this niche to maintain their stemness. And if they have no contact to the niche, they either die or differentiate. So this is the idea. So we consider for the moment we have a niche with fixed capacity. We are now trying to look into what happens if the niche capacity changes with age, but that's a different story. Then we assume that healthy and leukemic cells reside in the same niche. And what we basically assume is that leukemic cells can dislodge healthy cells from the stem cell niche, which in the end gives them a competitive advantage. So this is the idea. We represent this as an ordinary differential equation. And then we couple the dynamics in the niche just with the standard multi-compartmental model, accounting for progenitors, precursors, and mature cells, or post-mitotic leukemic cells. Then we do the same kind of sensitivity analysis. We see parameters of these cells do not matter so much. And then everything boils down that 2 parameters are very important. These are the proliferation of the stem cells, and basically the competitive advantage, if you like, so the probability with which leukemic stem cells can dislodge healthy stem cells from the niche.
So the competition is entirely in the stem cells in this model, and progenitors, do they compete with each other?
No, so what we have is we have a feedback signal such that if there is a lack of mature cells, all the cells can divide faster. But the competitive mechanism here is just in the niche.
So what is the term that mathematically
sorry, the computational dislodgement?
So what we basically assume is we have a niche, and then if a stem cell divides, one offspring stays in the niche, the other tries to dislodge another offspring. So it's basically it's a term depending on the abundance of healthy stem cells in the niche. So it is basically kind of this term where the more healthy cells are in the niche, the higher the probability that a leukemic cell can dislodge one of them. And then we look how to do these two parameters. So stem cell proliferation, dislodgement probability, which in the end is something like the self-renewal probability, because only if you can dislodge a cell from the niche, you can stay there and self-renew. How do they impact the speed of disease progression? So we see if the probability that healthy cells get kicked out of the niche is small, the disease progression, so here starting the simulation from healthy equilibrium, a small number of stem cells is relatively slow. Then if we have the very opposite scenario, if this dislodgement probability is very high, then the disease progression is also slow, which basically comes from the fact that stem cells always produce stem cells. If stem cells divide once to twice per year, so it takes quite a couple of time until the system really sees big amounts of cancer cells. And then the intermediate scenario, we see that the disease progression is faster. And then a question would be, can we extract such information from clinical data? So for this purpose, we used a clinical cohort, which was designed in Heidelberg. And the idea was to measure the blasts, so the cancer cell burden, and at the same time, because this is a bit more feasible, to quantify the healthy stem cells. This does not work in all leukemic patients, but in most of them. And then we used the data from diagnosis, namely the cancer cell burden and an estimator of healthy stem cells and very immature healthy cells. Then based on these, we tried to estimate proliferation and dislodgement probability. And based on the estimated parameters, just from diagnosis to cell populations, we tried to predict the speed of disease progression, and we compared it with follow-up data, where, for example, we checked 3 years later of do the other patients still alive, have they relapsed in meantime or not. And what we basically see is, in principle, this works. So again, retrospective data, one trial cohort, we did some cross-validation, it's not prospectively validated. That's still missing. And we basically see when the model predicts good survival, then patients survive longer, and in the opposite case, the opposite. And then the next question, could such a thing potentially help us in the clinics? You might all know there exist risk stratification schemes which are used in the clinics, which depend on mutations and cytogenetic risk scores. And what you often see in the clinic, there is this low-risk group where patients usually do well. There is the high-risk group where patients usually do worse or very poor, unfortunately. And there is a relatively significant group of patients with intermediate risk. And it is a very well-known problem that one part of these intermediate risk patients more or less behaves like the low risk, and the other part behaves like the high risk. But you don't really know who behaves how. And therefore, one thing which could be useful is to search for tools to allow you to further substratify this group of intermediate risk. And then we applied the same model procedure to patients with intermediate risk. And what you see here is basically that also in the intermediate risk groups, it works. So this means this modeling approach can help you to substratify patients which are at the moment put into the same risk group by clinicians. skip this. And so this means the model-based quantifications and simulation might provide you additional insights into disease progression, and it might help to really measure not only the general cancer burden, but also to try to quantify the immature cells at the time of diagnosis, because this might potentially help you to better stratify the patients and, for example, to decide who should get a closer follow-up and so on. And then what, of course, all of us know is that cancer cells are very heterogeneous. So now I go back to a slightly simpler model. I drop again the niche, and I want to better understand which parameters in our model drive the clonal selection process. So what I look at is a relatively simple framework. I have an arbitrary but fixed number of clones, one healthy cell lineage, and n leukemic clones, here each represented by two compartments, just mitotic and post-mitotic cells. The mitotic cells, again, characterized by proliferation rate and fraction of self-renewal. And then what we do is we start in the healthy equilibrium, and then we put, here is a simple example, the same number of mutated cells of each clone. So here you see we have in this simulations 4 different clones, each 1 cell per kilo of body weight at time 0, and then we let the system evolve. And this can us give already complex dynamics, which means some clones increase and then decrease over time, and others continuously but slowly increase. So this is just a system where we have clonal selection. All clones are there at identical proportions at time 0, no mutations, just feedback and healthy cells. And this is what we get. Then we can look at the same on a longer time scale. So here it's basically 2 years, here it's 5 years. And then we really see that the long-term dynamics can be very different from the short-term dynamics. So this means clonal competition is a potential mechanism which can explain why clones increase and then again decrease over time. So what drives this mechanism? Numerically, we see the higher the self-renewal of the leukemic cells, the longer the clone persists. And in the end, if we wait for t to infinity, only the clone with maximal self-renewal will survive. So Anna and Piotr Gwiazda and Jan-Erik Busse have proved this rigorously. So in this type of models, the self-renewal probability is the key parameter of clonal selection. Then this model has some other nice properties. So for example, what we also see is, as we observe in the clinics, if we simulate chemotherapy, so a very simple chemotherapy, just assuming that the quicker the cells proliferate, the higher the probability that they are killed. So epigenetics at the moment neglected. We are working on this at the moment. And what we see is first that even in absence of mutation, just due to selection, the relapse can be driven by different clones than the initial diagnosis. And what we also see is if we apply the same therapy after relapse compared to diagnosis, then the disease-free interval becomes shorter and shorter, which means the original treatment does not work anymore and the disease becomes, in this sense, more aggressive. So this model, also simple, can capture some features which we really observe in the clinics. Then a next obvious step would be to take this model of selection and now add mutations, because this would give us the chance to also have new clones appearing during the course of the disease. And what we did here is basically some mixed approach. We again represented each clone by 2 ordinary differential equations. So we started with a healthy cell and one founder clone. And then basically we assumed, which is a strong assumption, but we made it, that mutations arise during division, which means we then added a Poisson process to obtain from this Poisson process, where the rate depends on the cells dividing, gives us discrete time points where we add new clones to the system. Then we let it evolve, which means we have basically a system of ordinary differential equations, and at discrete time points, we add new clones and new equations. So this is the idea. And then we neglected neutral mutations. So what we assumed is that just in this model, to keep it simple, with each mutation, we consider the properties of the clone change. And I mean, if it would be a neutral mutation, we just wouldn't count it and would leave the clone in its, the new cell in its original population. And then we basically assumed that with the mutation, self-renewal probability and proliferation rate can change. And this is relatively interesting because then we know that there is clonal competition and that basically the selective mechanism can be exerted, for example, either through a niche or through a feedback. And then the question would be, how does the specific selective mechanism impact on the properties of the clones which get selected? So then we ran lots of different simulations of this model, and we looked basically clones at the top of the hierarchy and clones at the bottom of the hierarchy. How do the cell properties change? And what we see is that in this model, the self-renewal increases with the depth of the hierarchy. And indeed, we know from our other simulations, high self-renewal should mean poor survival. So one conjecture would then be the deeper the hierarchy, the poorer the prognosis. And interestingly, one group found 1 or 2 years ago that these linear hierarchies really seem to have a poorer prognosis compared to these branching hierarchies. So this means the topology of these phylogenetic trees might really coincide with some prognostic insights. Okay, then maybe in the last 5 minutes, I will come to clonal hematopoiesis. So this is now the bridge back to Morten's talk. We know clonal hematopoiesis is a phenomenon of the elderly. The incidence increases with age. And a big problem is that the mutated clones can transform into malignancies. Clonal hematopoiesis is linked to a high comorbidity burden in case of cardiovascular diseases, other inflammatory complications, and so on. And maybe the biggest problem is that in many respects, it is at the moment unclear how to clinically deal with this phenomenon. For example, should people with clonal hematopoiesis be excluded from stem cell donation? It's at the moment not known, and there are different studies pointing in different directions. And what I want to understand, similar as Morten, Jordan, and many others in the field, here are some references, is what governs the dynamics of these pre-malignant clones. Actually, if you look at data, so this is data from literature, the dynamics is much more heterogeneous compared to what we see in AML. So this is data taken from the paper by Robertson and Udin, and what you see is that even if you have mutations in the same driver gene, in some people, they increase over time, in some people they decrease over time, and in some people they more or less stay constant. So the question is, where could this come from? There are several ways. Immune effects might be one way. As Dominic said, the cell in which the mutations happen might be one explanation. A third explanation might be chronic inflammation. So we all know that at least in average, the level of chronic inflammation increases with age. We know that also in some of the CHIP patients, if you look at the expression of pro-inflammatory genes in the mature cells, it is increased. So this means there can be a feedback, not only in MPN, where this is relatively well understood, but also in CHIP, where basically the mutated cells increase the level of systemic inflammation, which in the end leads to a positive feedback. And we also know that chronic inflammation in mice and zebrafish, or maybe in Medaka, I don't know, at least in fish, can reduce the self-renewal probability of the wild type cells. So this means there multiple lines of evidence converge to the meaning of chronic inflammation in clonal hematopoiesis. We have different candidate models for this. So one model similar to the leukemic model, where just the mutated cells have a fixed competitive advantage. Then one model where the mutated cells trigger chronic inflammation and increasing their own competitive advantage. One model where mutated cells trigger chronic inflammation and decrease the competitive fitness of the healthy cells. And what we see is, with the limited data we have from literature, the longitudinal data of clonal hematopoiesis, basically the models can, what we see. So these are data points taken from Van Seventer. All three models can more or less explain this very sparse data, which is not so bad. The big problem is what all these models cannot explain actually is how the clones can decline again, unless we assume that stem cells happen, that mutations happen in a non-stem cell. Then it depends a lot on the parameterization if you can really cover and reproduce the time frame. So therefore, one thing which could bring non-monotonous dynamics could be that there are other independent inflammatory processes in the body, like inflammaging, like tooth infections, whatever. And then we, of course, can add this in the model that there is, let's say, some type of inflammation coming, not triggered by CHIP, but acting on the clones. And in such a system, if we assume just an arbitrary inflammatory signal, which can, for example, increase with age and then drop if you take in some medication or whatever, this could explain the non-monotonous dynamics as a proof of principle. Of course, the drawback is if you have an unknown signal which you cannot measure and which you can choose as you please, you can most probably recover everything. So that's the drawback. So as Morten said, we now try to measure cytokines and MPNs to get a better view on this. But one last point I want to make is there is one scenario where indeed basically all hematopoietic signals which you can imagine are perturbed at a defined time point, and this is bone marrow transplantation. So we know if you transplant patients, basically all the inflammatory signals, growth factors increase. So then the question is, how do CHIP clones respond to transplantation? And indeed, there is data for this. And here you see simulations of two versions of the model. One simulation is basically similar to our leukemic model. So what you see here is the variant allele frequency in peripheral blood, the point in time. The black diamond is the variant allele frequency in the transplant. So this is literature data, which I will show you later on. And then we see if we assume there is a fixed growth advantage, we see more or less a continuous increase of the variant allele frequency after transplantation. If you now assume that the competitive advantage of the CHIP clones is context sensitive, which means it is there if there are specific signals, it is not there if these signals are not there. So then we see, and the model would predict, a steep increase of the variant allele frequency in the year after transplantation, and then more or less a constant variant allele frequency. And if we now check what we observed in data. So this is data taken from Frick et al. So we really see in patients, unfortunately, again, there are multiple of such patterns. I only took out 2. We can see both. In some, we have an increase over many years, which would fit to the idea that the mutated clones have a constant advantage. But in other patients, we see there is an increase within the first year after transplantation, and then more or less it is constant. But this at least could be seen as a hint that there might exist patients where really the competitive advantage depends on the inflammatory and signaling environment and might not just be constant in time, but really requires some signals for the clones to grow and goes away if these signals do not grow. Then I stop here, not to be too much over time. I hope I have convinced you that disease dynamics are very complex, that it is challenging to really get good risk stratification and to try to predict the disease dynamics, and that especially before the overt cancer arises, there are many fluctuations, many inter-individual differences, which are not so well understood, and different models can potentially explain them. Then thank you for your attention.